What's up, Ninja Nerds? In this video, we're going to be talking about acute disseminating encephalomyelitis, abbreviated ADEM. I'm only going to write that thing out one time and one time only. The rest of the video is going to be all abbreviations. But before we get started on this video, please, if you benefit from it, you like it, hit that like button, comment down in the comments section, and most importantly, subscribe. So let's get into ADEM. All right, so let's start talking about the etiology, the pathophysiology of ADEM, acute disseminating encephalomyelitis, right? So we don't know the exact pinpoint causes, but we have some things that we believe could be a potential trigger or related to ADEM. So there's two components, because it is believed to be somewhat of an autoimmune type of disease. It's kind of like multiple sclerosis, just a really intense acute flare-up, and it tends to happen in younger individuals. Okay, so what are the environmental factors to this autoimmune disease? Well, it tends to be viral infections that are related here. And so there's two particular things. One is it could be an infection related to measles, mumps, and rubella. On the very rare occasion, it's rare, so I'm going to write that down, sometimes it could be from the MMR vaccine, okay? So either measles, mumps, rubella, or the vaccine against measles, mumps, and rubella could be a potential etiological factor. The other thing is a particular bacteria known as Mycoplasma pneumoniae, right? And the last bacteria that we want to be thinking about here is a very interesting one called Streptococcus beta hemolyticus. Okay, so Streptococcus beta hemolyticus types of infections. Either way, these are the pathogenic or infectious etiologies. There also has to be a genetic component, because this is a very similar type of autoimmune disorder like multiple sclerosis. Now, in multiple sclerosis, there was a particular susceptibility gene. We don't have a well-documented type of susceptibility genes, but there may be some type of susceptibility genes present, but just not known, documented, or elucidated yet. The other thing is that we tend to see this in younger individuals. So this tends to be something that we see in younger patients. Okay, so younger patients, we tend to see this more particularly. Now, how does this happen? It's kind of a combination of the two. So let's say here that at some point in time in life, we've been exposed to a particular pathogen. Okay, and we're going to represent a piece of that pathogen as this blue little molecule here, which we're going to call an antigen. So this is a particular protein component that is present on one of these pathogens. Our immune system encounters that and undergoes a process called phagocytosis. So it engulfs that. So here's an antigen presenting cell, and this one just so happens to be a macrophage. It engulfs the antigen. When it engulfs the antigen, it exposes a piece of that antigen on its cell surface in the form of what's called an MHC2 complex. Now, normally, it'll put this antigen here on that MHC2 complex. And then what it'll do is, it'll present this to another particular molecule, another particular type of cell. It'll present this to what's called a T cell, a helper T cell particularly. So here we're going to draw a helper T cell. And this helper T cell has a very specific type of protein here called a TCR, a T cell receptor. And what it'll do is it'll interact with this antigen presenting cell, the macrophage in this case, and it'll interact with that antigen that it expresses. So here we're going to have the MHC2 complex, and then here presented on that MHC2 complex is going to be the antigen. And so this will activate that T cell, because you know there's also another protein that comes over and extends and interacts here. And this is the CD4 protein. Once this happens, this becomes activated and may start proliferating and making more other different types of T cells with T cell receptors that will recognize that type of blue antigen. Now, here's what's really interesting. If you take this blue antigen from one of these pathogens and you compare that to the proteins that are actually within the myelin sheath, particularly maybe like the myelin basic protein, they somewhat look almost similar. And so what happens is our T cells may think that, oh, this is the antigen that we were exposed to in the past, that's foreign, but it looks just like this myelin protein. I need to attack the myelin protein. And so this type of thing here is called molecular mimicry. Okay, and so this can happen with the antigen looking like maybe the myelin basic protein, which is in the myelin sheaths, okay? All right, so let's talk about how that happens. So we know infections, particularly measles, mumps, rubella, on the rare occasion, the vaccine, mycoplasma pneumonia, streptococcus beta hemolyticus tend to be the environmental agents. The genetic, there's not documented susceptibility genes, but may be present, and we see this in younger patients. When exposed to this, they develop these immune cells that now have this molecular mimic reaction. How does that take place? Let's explain that. Here we're gonna have these T cells, right? These T cells that have pretty much kind of been primed in a way. What they're going to do is they're going to pass through the blood-brain barrier. You remember there's different components of the blood-brain barrier, right? The endothelial lining, the basal lamina, and the astrocytes. So generally the blood-brain barrier is very selective and it only allows for particular things to pass by. But what happens is these T cells have the ability to cross the blood-brain barrier and come into the area where the neural tissues are. When they come into this area, something very, very cool happens. They come and notice particular proteins that are present on the myelin and say, hey, this is really similar to the proteins that I was exposed to in the past via that molecular mimicry reaction we just talked about. When it does that, it becomes activated. When it becomes activated, it starts to release a bunch of different cytokines. These cytokines that it releases triggers a massive inflammatory reaction, leads to various effects. What are these effects? Well, first, the cytokines that I want you to remember, interleukin 1, interleukin 6, TNF alpha. There's another one that we'll talk about next, and this is called interferon gamma. Now, what do these things do? Well, the first thing I want you to know is that interleukin 1, 6, and 2 minocrotic factor alpha act on the endothelial cells and say, hey, endothelial cells, I want you to express a lot of cell adhesion molecules so that whenever white blood cells are floating through this area, they'll stick to you and then come into this area where there's a lot of inflammation going on. The second thing that it'll do, so that's the first thing. The first thing is it'll trigger these cell adhesion molecules to be formed. The second thing it'll do is it'll also tell the blood vessels to start dilating, to kind of dilate, and when you undergo this process called vasodilation, it increases the blood flow through the vessels. So more blood's going to run through this area, and if you bring more blood, you bring more white blood cells. More white blood cells can infiltrate into this area. Another thing it does is it also causes these vessels to become a little bit more leaky so that more things can leak out of the vessels, like proteins and different types of antibodies and different types of immune system cells. And so this is going to be increasing capillary permeability. So far we have three particular factors that are occurring. Second is the vasodilation. The third thing is the increased capillary permeability. The fourth thing is that some of these cytokines can get out here into the blood and activate other types of immune system cells. Maybe it'll activate B cells and more T cells to start to come to this area. What is that called? What is that fourth thing that it can trigger? The fourth thing that it can trigger is a process called chemotaxis. There's another thing. You know macrophages? Macrophages love this particular molecule. So macrophages may be coming to this area. And when macrophages come to this area, they can also be triggered by this chemotactic mechanism. But they really heavily respond to interferon gamma. So whenever there's a lot of interferon gamma, it activates, it stimulates these macrophages and triggers them to come out into this area. When the macrophages come out into this area, they're going to be starting to get involved in this kind of demyelinating and destructive process. So far, what do we have here? We have a fifth event here, and this fifth event here is going to be stimulating what's called your macrophages. So we're going to stimulate the macrophages to come to the area. So far we've had increased leukocyte, increase the cell adhesion expression, vasodilate, increase capillary permeability, trigger chemotaxis, activate macrophages to come to the area. Now, let's say once all of that happens and all these cells come to this area, what does it look like? So now, we're going to have some of these B cells come into the area, right? We're going to have some of these B cells come into the area, and these B cells, they're going to start getting activated, right? Because there's a lot of inflammation, a lot of cytokines. They're going to get converted into a very specialized antibody factory, and they're going to start popping out antibodies like IgG antibodies. And these IgG antibodies are going to be going and tagging these oligodendrocytes and the different components on the myelin that resembles that initial antigen via the molecular mimicry reaction. When it does that, it tags them, right? So this is the sixth thing to happen. The sixth thing to happen that I want you to remember is that there's going to be an increase in antibody production. So this is your antibody. You're going to have lots of IgG antibody production. When the antibodies are being produced, it tags this. And then that's when the macrophages say, you look good, oligodendrocytes. I'm going to come and eat you up. And so it comes and undergoes this process called phagocytosis. And so the seventh event that will occur here is you'll have phagocytosis of these oligodendrocytes and the myelin sheaths. And there's so much inflammation that not only will it destroy the myelin and the oligodendrocytes, but it may also involve some of the neuron itself. So you may get white matter destruction and gray matter destruction. So you may injure the neuron and also injure the oligodendrocytes that myelinate these axons, okay? So once that happens and you destroy and injure the myelin sheaths and even injure the axons, what happens is later you're going to have a lot of these plaques that will start to form. Okay, so you're going to start forming a lot of these nice little plaques that will form on the axons of these neurons. And you may even have some slight injury to the axons or some actual injury to the neurons themselves. Now, T regulatory T cells may come in and get involved in this process, but usually acute disseminating encephalomyelitis, it's such an intense type of reaction, and it usually occurs acutely that it's not going to happen anytime soon whenever they present to you, okay? So they're not going to have this kind of like relapse and then a remission by the time they've come and seen you. They've had this extensive demyelinating and inflammatory destruction that leads to clinical features, and then they will present. This leads to this condition that I'm only going to write once called acute disseminating encephalomyelitis. You guys see why now. I'm not going to write this again. That's a heck of a thing. So we also, again, abbreviate this ADEM. Okay, so this is a pretty intense kind of condition that can kind of come on very acutely, and again, something that you'll see generally in younger individuals who had a viral infection, a previous kind of vaccination, and then they'll present with a significant amount of neurodeficits that let's go talk about now. So now let's talk about the clinical features of ADEM. Now some of you guys might be saying, dang, Zach, I mean, this disease kind of sounds like multiple sclerosis in a way. It's kind of the same kind of molecular mimicry, same kind of like pathophysiology. Is it the same? It kind of is the same, right? Except one of the big things is that someone who has multiple sclerosis tends to be 20 to 40, tends to be female, and they also have these relapses and remitting types of occasions, right? With acute disseminating encephalomyelitis, we see this commonly in younger children, okay, adolescents, and it tends to be this single kind of one-time event. There's not an event and then a remission. It's this one-time event. And with this, there's also a couple other distinguishing clinical features that we'll talk about. All right, one of the first ones is that whenever there's exposure to these particular pathogens, right, so some of those viruses, the Mycoplasma pneumonia, the Streptococcus pneumonia, what that can do is that can activate and react with our immune system, right? And whenever our immune system gets involved, there was that trigger, that massive trigger or release of particular types of cytokines, such as interleukin-1, such as interleukin-6. And what these were doing, and even tumor necrotic factor alpha, these were acting on the hypothalamus. And you know what's in the hypothalamus? They trigger prostaglandin E2 production, which resets the hypothalamic thermostat. And as a result of that, triggers an increase in your body temperature and leads to fever. So sometimes in people with acute disseminating encephalomyelitis, they can present with fever. And they may even present with other like mild symptoms such as headache and maybe even some nausea, okay? So that's the first one that kind of helps a little bit with differentiating. Single time event, younger children, fever, headache. They do present with optic neuritis though, so they can really inflame that cranial nerve 2 and affect the visual pathway. And so they could develop optic neuritis. And again, with optic neuritis, they can have decreased visual acuity. And they maybe even have some color vision abnormalities. So we'll put abnormalities, okay? The next thing is, again, go back to your things here where you have these motor neurons that are located within the primary motor cortex, the premotor cortex, and they give all their axons that run down via the corona radiata, internal capsule. They come down through the midbrain and they give off these fibers to cranial nerves, and particular cranial nerves at that. What are these cranial nerves that I want you guys to remember? Well, it can innervate cranial nerve 5, which is the trigeminal nerve. It can innervate another one, which is the 7th cranial nerve, known as facial nerve. It can innervate this blue one. It's actually called the nucleus ambiguus, but that is involved in particular fibers of cranial nerves 9, glossopharyngeal, cranial nerve 10, vagus, and cranial nerve 11, the accessory nerve. And then last but not least, it can also innervate the nucleus of cranial nerve 12, the hypoglossal nerve. Now think about this. And a person who has some type of acute disseminating encephalomyelitis, not only do you demyelinate oligodendrocytes in cranial nerve 2, but you can demyelinate and destroy the oligodendrocytes that myelinate the axons of the corticobulbar tract. When you demyelinate those axons, it can present as a very type of particular condition, which we refer to as pseudobulbar palsy. Okay, pseudobulbar palsy. Now, how does pseudobulbar palsy present? Let's not get too crazy. Let's think very simply. We demyelinate these corticobulbar tracts. This is an upper motor neuron type of lesion. If you're not supplying or stimulating cranial nerve 5, that goes to the mastication muscles, your chewing muscles. There'll be decreased chewing. And again, it is an upper motor neuron lesion, so if you did the jaw jerk reflex, it would be brisk or hyperactive. It also supplies cranial nerve 7. So if you affect this one, you could develop absent facial expressions. The other thing is it can hit the inhibit the involvement of cranial nerves 9, 10, and 11, which are involved in speech production as well as involved in swallowing. And so if these are injured, it can lead to dysphonia, and it can also lead to dysphagia. And then last but not least, if it involves the actual 12th cranial nerve, the hypoglossal nerve, this may also be involved in speech production, but particularly the articulation of speech. So there may be some type of dysarthria present. Okay, so we can have fever, headache, optic neuritis, pseudobulbar palsy. This next one is the big one and most important one to remember, involved in the differentiation. There's a lot of demyelination of the higher order brain centers. And whenever you injure these, it really affects a particular mental status and leads to encephalopathy. So these individuals can present with what's called an acute an altered mental status. So they can present with an altered mental status, so they can present encephalopathic, maybe very irritable, maybe very confused, right? So they can present with an altered mental status. We can kind of say encephalopathy. Very big one. I really don't want you guys to forget this. This is one of the key features that distinguishes them. The other one, son of a gun, is it can involve the reticular formation. So it can demyelinate some of the axons within the reticular formation. What is the reticular formation involved in? It takes sensory information from your body, whether it be the sensations, auditory pathway, visual pathway, all of that stuff goes to the reticular formation, and from here it sends it up to your thalamus, and then from your thalamus it goes to the cerebral cortex. Well, if we demyelinate that, we affect the arousal of the central nervous system, and these can actually lead to decreasing level of consciousness, sometimes worst case scenario, comatose, right? and worst case scenario, but usually it's a declining level of consciousness. The other thing is it can demyelinate some of the axons that are involved within the cerebellum. If you hit these axons, this can lead to ataxia. And in some cases, it may even lead to seizures if you involve a lot of the cortical neurons. Okay, so we have fever, headache, optic neuritis, pseudobulbar palsy, big thing here, encephalopathic. What else could it hit? We hit kind of the brain stem. It also extends down into the spinal cord. So what aspects of the spinal cord? All right, so next thing is if we injure some of the actual structures, the tracts within inside of the spinal cord, there can be some really problematic things. So let's think about this. What is this column here called? Your dorsal column. What does the dorsal column do? The dorsal column is involved in sensations, particularly like proprioceptive sensations, fine touch, discriminative touch, a vibration sense. So if you demyelinate those axons, you can have decreased proprioceptive, fine touch, discriminative touch, and vibration sensations. If you injure these red tracts, which are your corticospinal tracts, your corticospinal tracts are upper motor neurons. They're going to give fibers that go to the lower motor neurons in the anterior gray horn. You injure that, you can develop upper motor neuron lesion symptoms. And this can present with what? It can present with weakness, obviously, and it's going to be more of a spastic type, right? So they're going to have increased tone. They're going to have increased deep tendon reflexes, also known as hyperreflexia. And they're even going to have positive pathological signs called a Babinski sign. Okay? Now, the next thing, what if we demyelinate the axons here within the anterior lateral spinal thalamic tract? Well, think about that. The anterior spinal thalamic tract is responsible for carrying crude touch and pressure. So there may be a decrease in crude touch, and there may be a decrease in pressure sensations that can be carried, right? It's super simple. We're cruising. Well, what about the lateral component? The lateral component carries pain and temperature, so there could be decreased pain and temperature sensations that are being carried as well. Man, we good. All right, what else? In certain scenarios, remember I told you that not only can it demyelinate the axons, but there also is the ability for it to damage the gray matter, some of the neuron. In some cases, it may even injure the anterior gray horn. And if it injures the anterior gray horn, it may present with a lower motor neuron lesion as well. And so this can also present with weakness, but this weakness tends to be more flaccid. So the tone is really decreased, hypotonia. The reflexes are decreased. They have hypoactive reflexes or hyporeflexia. They don't have a positive pathological reflex. They actually have these like little weird twitching of the muscle, and these are called fasciculations. Okay, so these tend to be the big clinical features that you see in ADEM. Now let's talk about diagnosis. All right, so at this point in time, now we're at the diagnosis. So at this point we know, okay, there was a particular trigger, maybe some type of viral infection or bacterial infection. They then acutely developed particular types of symptoms, right? And these symptoms tended to be some weakness. So we talked about the particular spinal cord lesions. We talked about the altered mental status, how that was a big defining factor, maybe even some fever, some headaches. You start thinking, okay, this is a first-time event, it's a child or it's an adolescent. I'm thinking ADEM. What is the first-line diagnostic test to ADEM? Well, I need to get an MRI, and I need to get an MRI of the brain and the spinal cord because this involves both of them. Right? We obviously see that by the clinical features above, but I want to see this with and without contrast. So when I do that, here's the big thing. We can see a lot of lesions in that area of the cortex. We can see a lot of lesions in the cerebellum. We can see a lot of lesions in the brainstem, and we can see a lot of lesions present within the spinal cord, just like an MS, right? Here's the big thing though. When you look at the actual supratentorial region, so supratentorium, okay? So more of the upper parts of the cerebral cortex. You tend to see a lot of demyelination, but it tends to be asymmetric. And we call it multifocal. So you see lots of demyelination in the supratentorial area, but it's not symmetric and they're all over the place. That's a big thing. You still see them in some of the same locations: cerebral cortex, brainstem, cerebellum, and spinal cord. The other thing is look at particular sequences, T1, your T2, particularly T2 flare, and look at them whenever you give contrast. The T1 tends to present as hypo-intense, meaning that it may show up dark, where there's demyelination. T2 tends to show up hyper-intense, which means it's going to show up bright wherever there is demyelination. And then the contrast tends to enhance particular acute lesions in this scenario, okay, which you'll see a lot of in ADEM. Okay? Now, what other tests could I order to aid or enhance the diagnostics involved in ADEM? I can obtain a lumbar puncture. Now, when you do a lumbar puncture, you're taking some cerebral spinal fluid, right? So you're going to take some of the CSF and you're going to analyze it. Well, think about the pathophysiology. What was one of the things that we saw a lot of? Well, we did produce a lot of antibodies, right? So there was a lot of like antibodies that were being produced. So you may see some increase in IgG antibodies. So like some oligoclonal kind of banding. So there may be lots of IgG antibodies in there. The second thing is there was lots of white blood cells, lots of B cells, lots of T cells, lots of macrophages that were invading into there. This is called pleocytosis. But there's one other thing. Since these viruses and infections can be involved in this process, we should take into consideration throwing out some cultures to test for Streptococcus beta hemolyticus, maybe mycoplasma, and maybe even run a viral panel to make sure that we're not missing any measles, mumps, or rubella as well. Okay? So that is what I want you guys to remember. Get an MRI of the brain and the spinal cord with or without contrast, look for hyperintense, hypointense lesions, and then get an LP to look for increased proteins, increased white cells, and then throw some cultures and viral panel in there to rule out any active infections. All right, so now we've diagnosed the ADEM. Okay, we got to treat. They say, okay, what's the first treatment that you want to give to this child who has ADEM? You start them on high-dose IV corticosteroids. So you're going to start them on high-dose corticosteroids. Now, how does this work? When you give a high-dose corticosteroid, you got to remember the kind of the mechanism of what we talked about before. Remember those T helper cells? They were going ham and just blasting out lots and lots of cytokines. And lots of these cytokines induced a lot of mechanisms like increasing vasodilation, increasing capillary permeability. They were causing chemotaxis. They were increasing the expression of those leukocyte adhesion molecules, activating macrophages, all of that stuff. And when they were doing all of that, what if we gave a drug, a nice drug that can really kind of shut down or decrease the activity or function of all of these cytokines? That's what high-dose corticosteroids will do. They'll shut down the inflammation to reduce a lot of the consistent damage that can occur in ADEM. What if that doesn't work though? What if you give the high-dose corticosteroids and it doesn't work or they have a contraindication to it? Let's try to clean the blood and get rid of all those nasty antibodies or proteins that may be the ones that are consistently causing this demyelination and this acute onset. And so how do we go about that? Well, generally, again, let's say that here your blood is kind of flowing through here and coming out here. Here's going to be some antibodies here, and maybe there's some other proteins that are involved in this inflammation. They're going to get pulled out via this tubing from the blood, and they're going to run through this chamber here. And what this chamber does is it clears out the antibodies and kind of puts them into this separate vial. So it's going to run through this chamber, get cleared. After it gets cleared, it's then going to run back to the blood vessel via this tubing. But on the way, it picks up clean, healthy plasma from a donor, or it's saline from with a little bit of albumin and other solutes that gets carried back through this tubing and back to the patient's blood so that you've cleared it of any problematic pathogens and given them good, clean type of plasma. So this could be a thing that we could do here, okay? Now, If we've tried this, right, we've given high-dose corticosteroids, we've given the second line, which is the plasmapheresis, another option that could be potentially helpful. It's not as great as plasmapheresis, but it could be an option instead of plasmapheresis. So you could do high-dose corticosteroids. That doesn't work, you have plasmapheresis. Or, remember how our plasma cells make antibodies? And these antibodies were responsible for tagging all of these Schwann cells, the oligodendrocytes and the myelin, and then enhance the macrophages to come and phagocytose them. I could give them another drug, which is called IVIG. It's basically an intravenous immunoglobulin, and what it'll do is it'll take and bind to these IgG antibodies and hinder them from being able to bind to the oligodendrocytes and the myelin. So you basically stop these antibodies from exerting their effect. Either one of these options can be considered. Now, one last thing that I want to mention before we end this video is that, remember I told you that this can present pretty significant, a single event. With having a fever, with having all of these weaknesses, you have to be on high alert and be thinking about, what if this is like a meningitis or encephalitis or a bacterial meningitis? You think about that if this is, regardless if it's a meningitis, encephalitis, bacterial, or viral, you need to treat them accordingly until you get the cultures back or your viral panel back. So remember, with this, you have to be thinking about potentially meningitis or encephalitis. So start them on antibiotics to cover for the bacterial infections, and then start them on antivirals until the cultures come back to cover for the viral infections. And once they do, you can discontinue these, okay? If it's clear of any type of particular infectious agent. All right, so that covers ADEM. All right, Ninja Nerds, in this video, we talk about acute disseminating encephalomyelitis. I hope it made sense. I hope that it helped. I hope that you guys liked it. As always, Ninja Nerds, until next time.